Hello everyone, I welcome you all in the series of lectures on biomolecule. Today we are going to discuss about the 10th lectures. Before going to the details of today's lecture, I would like to give a recap of last lecture. In the last lecture, we talked about the structure of proteins, and there we discussed about the primary structure, secondary structure, tertiary structure and quaternary structure. In today's lecture, we will talk about enzymes, another biomolecule enzyme. So, let us discuss about the enzymes. Enzymes. What are enzymes? Essentially, all organic reactions that occur in cell in cells require a catalyst. So, essentially basically catalyst, you all are familiar that when we talk about a chemical transformation, when two reactants react with each other and lead to the product, generally to push this reaction in forward direction or to happen this reaction, we use another entity which promotes this process, and that is called as catalyst. Similarly, all organic reaction whichever happens in the cells are getting are required to have a catalyst, and these biological catalyst, most biological catalyst are called enzymes, and these enzymes are which are globular proteins, Each biological reaction is catalyzed by a different enzyme. So, they are very specific. Each biological reaction is catalyzed by a different enzyme. So, to make it in the equation form, I will so substrate is getting converted to product. As we talk about chemical transformation, so here also it requires enzyme. How things happen in the biological system? See, in the chemical reaction, what do? We take two reactants and there we add catalyst in the same one part and things happen. It leads to the product formation. How things happen in the biological system? In the biological system, basically substrate reacts at the active site of enzyme, and that enzyme active site brings two reactant in the vicinity, and that closenates lead to the product formation, where they can interact with each other. So, the enzyme binds its substrate at its active site. A pocket in the cleft of the enzyme. What is active site? Basically, active site, a pocket in the cleft of the enzyme. Cleft of the enzyme. So, let me make the structure of enzyme. So, this is a structure of enzyme. And as we are talking about the cleft, this is the cleft. So, enzyme, and this is the active site, this cleft where the substrate is bound at the active site basically. The substrate is bound at the active site. Active site. Now, the specificity of an enzyme, how, as I mentioned, I would like to mention here that enzymes are very specific. So, if you have one enzyme, it will not work for all the transformation. For one particular transformation, only one enzyme is there. And how do they attain this specificity? So, the specificity of an enzyme results from the conformation. Results from its conformation and the particular amino acid side chain. Particular amino acid side chains that are at the active sites. So, this is the active site I have made, and they kind of fold in a specific fashion, and that way they make a particular I can say the cleft or groove where this substrate particular substrate will go and react at the active site. So, the specificity of an enzyme results from its conformation, it attains a particular conformation and the particular amino acid, because that particular amino acid will be responsible for the binding. So, amino acid and its conformation, both factors are there. Particular amino acid sites and side chains that are at the active site. At the active site. For example, an amino acid with a negatively charged side chain an amino acid with a negatively charged side chain negatively charged side chain at the active site can bind with a substrate which has a positively charged group, which has a positively charged group. Also, here hydrogen bond interactions are possible. So, hydrogen bond donor with hydrogen bond acceptor on the substrate. Here, this is at the active site, hydrogen bond donor with hydrogen bond acceptor. And a hydrophobic amino acid side chain hydrophobic amino acid side chain can associate with hydrophobic groups on the substrate, can associate with hydrophobic groups on the substrate. Hydrophobic groups on the substrate. And to explain all these interaction, Emil Fischer proposed the lock and key model. Emil Fischer proposed the lock and key model. Lock and key model. Lock and key model to account for the specificity of enzyme. For the specificity of an enzyme for its substrate. Enzyme for its substrate. So, let me represent it in a pictorial way. So, I will first draw lock and key model. So, this is basically I am making this active site of this enzyme. Now, you can see here it has specificity. The groups of the active site only can fit to the substrate, and this can lead to the complex. Now, this you can see that substrate binds with the enzyme active site and make the enzyme substrate complex. Okay. This is called lock and key model. This is very old, 1894 it was proposed by Emil Fischer. Half a century back there was another model came in existence, and that is called induced fit model. In induced fit model, basically it was proposed that the substrate comes vicinity of the enzyme, and even if it does not fit to the active site, through the interaction, slight interaction, it will start and slowly it will get fit to the active site. So, to make it that to understand, I will again read one more pictorial presentation. So, this is a enzyme. Now, if you look over the structure of active site, and let me draw the substrate. Substrate is same. So, now the substrate does not fit, here it does not have this active site is not matching, one is matching, another is not matching. However, when they come in vicinity, this interaction leads to the so, now it gets fit actually, and this is called induced, because after coming in vicinity this substrate is able to fit with the enzyme, and this model is called basically induced fit model. So, this is induced fit model, and this was presented in 1958 by Daniel and Cosland. This is called induced fit model. Now, we will talk that how catalyst, basically the enzyme catalyzing the reactions, how do they catalyze, basically catalyzing the reaction. So, how do they catalyze? Let us talk about that, how the enzyme works. So, some of the amino acid side chains are acidic, some are basic, and some are nucleophilic. And because of this characteristic, they portray that kind of interaction with the substrate. So, as I mentioned that some of the amino acid side chains, as we know that enzymes are made up of amino acids. Okay. So, basically it is made up of peptide, and peptide chains are made up of amino acid. So, some of the amino acid side chains of the enzyme serve as acid, base, and nucleophilic catalyst. Nucleophilic catalyst. And many enzymes also have metal ions at their active site. And many enzymes have metal ions at their active site. That act as a catalyst. That act as catalyst. These species are positioned relatively to the substrate precisely where they are needed for catalysis. Basically, these should not be embedded inside. These substituent should be positioned at the outer periphery where they can get exposed to the substrate, and so that they can act precisely. So, and because of the availability of these substituent, they inherit that nature, the whole active site start behaving like that, and that leads to the particular catalysis, particularly if it has acid substituent on the amino acid side chain, it will show that particular acid catalysis. If it has base at the side chain, then it can show basic catalysis. So, this factor is analogous to the way intramolecular catalysis intramolecular catalysis, intramolecular catalysis by acids, bases, nucleophiles, and metal ions. So, let me repeat again how the enzyme catalyzes the reaction. Basically, some of the amino acid side chain of the enzyme serve as a acid or base or nucleophile catalyst, and even many enzymes have metal ions at their active site, and these are the functional group or the metal ions are responsible for the catalysis, because the enzymes are made up of these amino acids, and these substituents are these species are positioned at the periphery of the these structures, particularly enzymes, so that it can get exposed to react with the substrate when the substrate comes in vicinity. And this whole thing, you can say that once the substrate comes in vicinity of the enzyme, it is almost like that how the reaction happens in a one entity, like that in a intermolecular fashion. You can say basically the intermolecular acid or base or nucleophilic or reaction with the metal ions, that leads to the enhancement in the rate of the particular transformation. So, if we talk individually about all these influencing factors, so an acid catalyst increase the rate of reaction. How does it do? Increases the rate of a reaction by donating a proton to the substrate. Proton to the substrate. A base catalyst increases the rate of a reaction by removing a proton from the substrate. Removing a proton from the substrate. A nucleophile catalyst increases the rate of reaction increases the rate of a reaction by forming a new covalent bond with the substrate. By forming a covalent bond with the substrate. amino is also one important information I will like to pass on, that amino acid side chains can stabilize transition state, can stabilize transition state by London dispersion forces, by London dispersion forces, electrostatic interaction and hydrogen bonding. And hydrogen bonding. Enzymes are generally named after the compound or class of compound upon which they work. So, how are they named? Enzymes are generally named after the compound or class of compound upon which they work. Compound upon which they work. Upon which they work. For example, the enzyme that catalyzes hydrolysis of maltose is called maltase. The enzyme that catalyzes hydrolysis of maltose into glucose is named as maltase. Is named as maltase. So, for I can write it in the equation form. So, if here is the maltose and in presence of maltase, it gets converted to 2 glucose molecule C6H12O6. It gets converted to 2 glucose molecule. Sometimes enzymes are also named after the class of transformation they perform. Sometimes enzymes are also named after the reaction. After the reaction where they are used. E.g., For example, enzymes which catalyzes the oxidation of one substrate which catalyze the oxidation of one substrate with the simultaneous reduction of another substrate, with the simultaneous reduction of another substrate are named as oxidoreductase enzymes. Oxidoreductase enzyme. So, this is called oxidoreductase enzyme. This much about the enzymes. So, we saw that enzymes are basically a which can be named after the class of substrate on which they react. For example, we saw that how it can hydrolyze maltose to the 2 unit of sucrose, 2 unit of glucose, and the enzyme is called maltase, and the type of reaction, if enzyme is working for the oxidation and the simultaneously another substrate is getting reduced. One substrate is getting oxidized, another substrate is getting reduced, then it is called oxidoreductase enzyme. So, I will like to stop here. Now, in the next class, I will start with the vitamins. Vitamins. We will start vitamins. That what are the vitamins basically, and their types and how do they work basically. Thank you very much for listening.